Students, as you know, paper 3 in A levels is a practical examination. In today's video, we will discuss food tests from the perspective of practical work and how they are performed in the P3 practical paper. So in this regard, first of all, we are going towards the first test, which is the Benedict test for reducing sugar. And students, you know, the Benedict test is performed with the help of a reagent, which is called as Benedict reagent. For this purpose, you need to take test tube for that solution which has been provided to you in the examination hall. So you will add up about 2 to 3 ml in the test tube, about 2 to 3 to of given solution. And then you will add the same quantity of the Benedict solution. Then you will heat it up into a water bath for about 80 to 90 degrees centigrade for about 5 minutes. Then you will see the color change pattern or the color changes from blue to green, yellow, orange, or brick red can be seen over there. As you know, the Benedict solution is of blue color. So the color changes will occur. Blue will change into green, and then green color will be changed into yellow, and yellow color will be changed into orange, and orange color will be changed into brick red. So the brick red color or the color change pattern will show the presence of reducing sugar in the given solution. If the color change will not occur, that it remains blue, it means that is the no reducing sugars. Otherwise, if the color changes occur, that is from blue to green, yellow, orange, and brick red, it shows the different percentage of reducing sugar present in that solution. So you can see this color change occur due to a reason. And what is that reason? That is the reducing sugar reduce the copper 2 ions in Benedict solutions to copper 1 oxide, because the Benedict solution contains blue copper 2 sulfate ions. But when we heat the reducing sugar, then in that case, what happens? The reducing sugar denotes the electrons to copper sulfate 2. So it reduces to copper 1 oxide. So that is the reason this color change occur as we perform the Benedict test for reducing sugar. Now, in the next slide, you are able to see the different color changes which occur during this whole process. That is the blue color. If blue color is there, it means there is no reducing sugar present. But you can see from green to yellow, it will show that traces of reducing sugar are present. It means it shows the different concentration, the color shows the different concentration of reducing sugar which is present in that solution. So traces of reducing sugar are present if the color changes from blue to green and yellow. When the orange color is there, then you can see that the moderate concentration of the reducing sugar is present in that solution, and brick red color shows the large amount of reducing sugar is present there. So in this way, I would like to say that the Benedict test shows us the presence or absence of the glucose in the given or the solution which has been given to test. So copper oxide is insoluble in water and appears as brick red precipitate. So the color changes depends on the concentration already told you. so that the blue color shows the different no percentage of or no concentration of sugar, and other color shows the different amount of reducible sugar present in the solution. Now I would like to move the second test, which is the Burette test. And this test is performed to find out the presence of proteins present in the given solution. So in this regards, first of all, you have to prepare the sample. That is, you need a test tube in which you will take the some amount or the of the given solution. If solution, or if you are going to test a solid food, then first of all you have to crush it and mix with distilled water. Why do you need to crush it? Because in this way you will increase its surface area. You will increase its surface area and the reaction will be fast. So then filter out the solid particles so you have a liquid extract. So when you will crush it, then you will mix it with distilled water, then it will be, you will be able to find out the percentage of amount of proteins into that distilled water. So then you will filter it out and then you can test it for the presence of proteins. Then you have to add the a reagent which is called as Burette reagent. So about 2 ml of your sample into a test tube. I have already shown you, add an equal volume of the Burette reagent, but you have to put it drop by drop, drop by drop. So with the help of a dropper, and then gently shake the test tube and let it stand for 5 to 10 minutes at room temperature. Then allow the color reaction to develop. So the color will be changed. If the color is changed from blue to purple, then it shows that protein is present in the given solution. Otherwise, if the color remains the same, that it remained blue, it means there is no proteins present in the given solution. Now you can see in this uh, slide that the blue color shows the that negative proteins are absent. No protein is present in, but when the color is changed from blue to deep purple, purple, so you can say the proteins are present in this solution. So what happens in the solution? The Burette reagent, it reacts with peptide bonds. So the Burette reagent, it will react with the peptide bonds in proteins, then it will produce a purple color. So the reason to producing a purple color is that proteins are present. So the Burette reagent will react with the peptide bonds and then it will produce a purple color. Now the next is ethanol emulsion test. Ethanol emulsion test is used to find out the presence of lipids, fats and oils into a given solution. So here in this case, what will happen? First of all, you have to prepare the sample, place a small amount of your food sample into a test tube. So you will add up into a test tube the sample solution. Then if it is a solid, again, if it is a solid, you have to crush it, grind it first, then you will take it into the test tube. Then you will add the ethanol into it. That is the same quantity of the ethanol. you will add it into it. Then you will shake the test tube thoroughly to ensure all lipids present completely dissolve into the ethanol. The fats are not dissolved into the water, but they are dissolved into the ethanol. So they will be dissolved. A clear solution will be there. So at that time, you can, this is very optional, that you can allow the mixture to settle for a minute, or you can decant the clear ethanol liquid into a new clean test tube, leaving behind any solid residue. It depends, but you can do without decanting it. Then you can see when you will add ethanol into the sample solution or into the sample solid which has been given at that time. So the clear solution will be appeared over there. Then you will add water. Add an equal volume of distilled water to the ethanol solution as you have added 2 ml of ethanol, and you will add the 2 ml of water into it, or distilled water into it. Then you will observe the result. You will shake it thoroughly and observe the mixture, and you will find out the appearance of cloudy, or you can say the cloudy appearance, or the appearance of the emulsion can be seen. So you can see on screen that when you will add water into it, that there will be the formation of emulsion. So what happens over here, that's, the sample solution dissolves into the ethanol. I would like to write over here as ethanol plus sample solution. They will make a clear solution, because this ethanol will dissolve the fats into it. But when you will add the water, plus add water, so what will happen? It will form the tiny droplets. Tiny droplets of lipids or fats, which will create a cloudy emulsion. It will create a cloudy emulsion. So this cloudy emulsion will show the presence of lipids into the given solution. If it does not happen, or if the cloudy emulsion is not formed, it shows the negative result for the fats or for the lipids. So if the cloudy appearance is there, you can see that the lipids are present in the given solution. Now, there is another test which is very important, and it is added, has been added into the many papers of the Cambridge. So non-reducing sugar test, first of all, again, we have to prepare it for, we have to make the sample solution. So take a fresh sample of the solution and add a few drops of dilute hydrochloric acid to it. First of all, what will you do? That is, there will be the sample solution. HCl, few drops, and after adding the few drops of HCl, you will heat it up. At that time when you will heat it up in a water bath, so what will happen? For a 2 to 3 minutes, acid will hydrolyze the glycosidic bond in the sugar. So the non-reducing sugar will change into the reducing sugar. So this neutralization allows the solution to cool, and carefully sodium hydrogen carbonate. Then you will add the hydrogen carbonate into it to provide a alkaline solution or to neutralize it. Because when you will add the HCl into it, it will become acidic. Then you have to neutralize it. For neutralization, you will add sodium hydrogen carbonate. So when acid, which is an alkali or which is an alkali or alkaline solution, then it will may change, it will neutralize, and then you can see it will change into the reducing sugar. Now, what will happen when it will be changed? How can you come to know that now the non-reducing sugar has been changed into the reducing sugar? For this purpose, you are going to conduct the Benedict test over it. Then you will do the Benedict test. For Benedict test, you will add then you will add Benedict solution. So you will perform the Benedict test again in the water bath, and as a result, you will come to know that what happens, that is the color changes occur again, that is from blue to green, green to yellow, yellow to orange, and orange to brick red. So this color pattern will show the presence of reducing sugar in that solution, which was firstly non-reducing sugar. Now we have changed it into the reducing sugar, and when we will apply the Benedict test on it, then it will give the results. That is, it can be changed into green color or yellow color or orange or brick red. But if it is changed into blue to green, it is a positive test. It is a positive result that the solution has been changed into reducing sugar from non-reducing sugar. Now the last test which we are going to discuss today is the iodine test for starch. So iodine test for starch, you can know that this is the confirmatory test to find out the presence of starch into any given solution. For the purpose, you will take the sample solution, you will add iodine into over it. So the when you will add the iodine into the solution, if the color is changed from this yellowish brown to yellowish brown to blue black, so it show the presence of starch into the given solution. If it will not, it is not going to change. If it remained the same, that is yellowish, because the color of iodine is yellowish brown. If the color remains the same, it shows that no starch is present in the given solution. So it was all about today's video on food test, and please like, share, and subscribe my channel to watch more videos on papers and on different topics which we will discuss now. Thanks for watching.